Hello. It's a real pleasure to connect with you at the meeting on the Mesa. As a former chairman of the board of ARM, it's always been my favorite meeting of the year, and I hope you're all well in this very unusual year of years. For those just getting to know AVRO, we're a leading lentiviral gene therapy company targeting multiple monogenic rare diseases. I remind you that this presentation contains forward-looking statements. Statements are subject to a number of risks and uncertainties, including those risk factors in our latest 10Q. Our mission at AVRO is simple: to enable individuals with genetic disease to live longer, better lives. We aim to halt disease progression with a single dose of gene therapy. And what that means to our patients is freedom from painful, debilitating symptoms and disease progression, but also freedom from the burden of lifelong biweekly infusions. We're now 12 patients into this journey and are very excited about the results that we have to share today. What we've done in the first 5 years as a company is built the leading lysosomal disorder franchise. In fact, each of these, Fabry, Gaucher, cystinosis, Pompe, are sister indications. And with proof of concept already established in Fabry disease, we believe that the probability of success in these sister indications with related biology is increased. We've currently dosed patients in 2 Fabry clinical studies, 1 cystinosis trial, and recently dosed our first Gaucher type 1 patient. Now, due to time constraints today, I will only focus on the Fabry disease clinical results. To orient you a little bit about these indications, they're large and they cost the healthcare system a fortune, hundreds of thousands of dollars a year. And in fact, there's an Erasmus publication that quantifies the lifetime cost for a Fabry patient between $12 and $14 million per patient. There's about 30,000 patients across these 4 diseases, and the original leading ERT companies, Shire and Genzyme, now Sanofi and Takeda, still sell roughly $4 billion a year in these indications. So the implication, of course, is that if we hit our target product profile of a one and done safe gene therapy, we believe we not only merit first-line status, but would save the healthcare system millions and millions of dollars for each and every patient treated. So why do we think gene therapy has a role where there already is an established standard of care? It comes down to pharmacokinetics. On the left, each of these blue spikes represents a patient visiting an infusion center, getting a 1 to 2 hour infusion of enzyme every 2 weeks for life. But the challenge is that if you test the patient's blood 12 hours post-infusion, the enzyme's gone. So right enzyme, bad pharmacokinetics, and the whole raison d'etre of gene therapy and of AVRO as a company is, what if you could deliver 24/7 expression of enzyme, of active protein, distributed through the body head to toe? The goal would be to untether patients from these biweekly infusions, but more important than that, to halt disease progression by bathing cells, tissues, organs, and enzyme, as opposed to just this stop-start. And that's what we set out to do. And this is our approach. This one ex vivo platform applies across all of the portfolio. It's autologous. We select the patient's CD34 hematopoietic stem cells, give them an upgrade with our proprietary 4-plasmid lentiviral vector system to deliver the healthy gene. The cryopreserved cells are then shipped to the clinic, and after a finite conditioning dose to create space in the bone marrow, the gene-modified cells are reinfused. So this is a very vanilla process. It's been validated and proven in hundreds of patients across multiple monogenic diseases. Towards the end of the presentation, I'll talk about how we at AVRO have optimized this. But just to orient you, this applies one-to-one across all of our diseases. Now, to jump right into our lead program, Fabry disease. Fabry disease is often thought of as impacting kidney and cardiac, but really it's a disease that manifests serious symptoms head to toe. Patients talk about peripheral pain, the feeling that their fingers are on fire, GI distress. They can tell you every bathroom from home to work. And importantly, central nervous system, what the patient groups call the Fabry fog, executive function deficit due to white matter lesions. Life is cut short 2 decades, but even everyday life is a challenge with Fabry disease. So a very serious monogenic disease. We are currently running 2 clinical trials, a fully recruited phase 1 and 4 out of 8 to 12 patient recruited already phase 2 trial. I'll start with just some early data of the phase 2. This is perhaps the most exciting and compelling data. The first patient to have reached the primary endpoint in the phase 1, in the phase 2, ERT naive patients. And here the endpoint is a kidney substrate biopsy. So the average number of GB3 inclusions per peritubular capillary. So right away, 87% is a very impressive number in terms of driving down the substrate, the fatty substance in the kidney. But perhaps more important than the 87% is the absolute level of 0.7, which is either at or darn close to full effective clearance. So we're very satisfied with this in terms of results, in terms of what it means to the mechanism of action, but it's also a very well-recognized primary endpoint, and the same methodology has served as the basis for registration for companies like Amicus with their chaperone therapy recently with FDA and EMA. Behind the kidney biopsy are a number of very well understood, validated biomarkers in this field. The Shires and Genzymes have spent 20 years building up, investing in this field. So there are some very well recognized biomarkers. For example, the alpha-galactosidase activity itself in both leukocytes and plasma. And here you see across all of the phase 2 patients is an absolute plateaued and steady 24/7 expression of enzyme, exactly what we hope to be able to demonstrate. So first point, we're very, very satisfied with the enzyme activity as measured in plasma and leukocyte. But of course, the eye is drawn to patient number 4, which has a higher level. And I'll get back to that. That's the first patient to receive our upgraded Plato platform. And I'll talk more about Plato towards the end. But the key point is all of the patients are expressing enzyme activity that we believe are at sufficient levels to control the disease, as measured by the lyso-Gb3 reductions in the plasma. Across all of the evaluable patients, we see a pretty significant 49 to 86% reduction at the most recent time points. I'd highlight that patient number 2 is a cardiac variant, a different mutation, which doesn't accumulate substrate in the plasma. So not included in this evaluation, but we included his numbers just for transparency's sake. So what we can show in the biomarkers are enzyme up and elevated and substrate toxic metabolite driven down as a result of this single infusion of a gene therapy. And then of course, a very hotly looked at measure in gene therapy is the vector copy number. And really what we hope to show is a consistent vector copy number plateauing over time, which is what we see. And again, that 4th patient receiving the Plato upgrades has a higher vector copy number. But I'd even call out, for example, patient number 2 in blue at 18 months with a vector copy number of 0.1 in vivo translates into 5 or 10% of all nucleated cells carrying 1 to 2 copies of the transgene. For an adult Fabry patient, that's 2 or 3 billion transduced leukocytes manufacturing and distributing protein through the body. So these levels of vector copy number have been shown to be sufficient to control the disease to date. Now, if I jump to our phase 1 study, and I'll just show you one slide which captures the results to date, which is, are these patients controlled as measured by their plasma lyso-Gb3 reduction? And in this study, these were ERT switch patients. So we have the benefit of looking in a within patient control to show how well were biweekly infusions of enzyme controlling the disease and how well is gene therapy able to do so. And gradually, patient by patient, they've had their ERT discontinued. So what we can say to date is that all patients on gene therapy are well controlled as measured by their plasma lyso-Gb3. 4 out of 5 are a significant reduction versus baseline, and even the 5th is absolutely in line with ERT levels. Net, gene therapy provided a 26% further reduction of lyso-Gb3 across all patients, and all patients who have had ERT discontinued remain off ERT. Very satisfying results. And then the final efficacy measure is something we've been waiting a long time to share, and we shared just in our recent quarter update in August of this year. And for perspective, kidney is one of the organs that gets impaired, and in the Fabry population, whether you're looking at natural history or actually patients on ERT, you see in multiple publications this very consistent, steady decline. So the slope of eGFR declines year on year. So just for perspective as a baseline, and now when you look at all of our patients in both phase 1 and phase 2, what you see is a different outcome. You see a stabilization of eGFR to date across all patients. The one patient that started with an eGFR at 48 does decline, you see on the bottom, but even that is at a rate of decline that is significantly less steep than comparable patients already starting with kidney disease at such a low level. So we see a different curve, a different slope, and in fact, to generalize, we see stabilization. We'll continue to monitor this because this is one critical measure to assess, are we realizing that initial mission of halting disease progression, encouraging eGFR results. Now, in terms of safety, there are no unexpected safety events or trends identified. In fact, the lenti approach is not impacted by the emerging safety signals with AAV gene therapy related to liver tox or immunogenicity. And in fact, we've seen no SAEs related to our gene therapy across any of our 4 studies. What is observed is a predictable set of adverse events and SAEs related to the concomitant conditioning regimen. We believe that they're transient and manageable, and I'll talk about this more in the subsequent Plato section. And I have to say, I can't update AVRO without talking about the immense efforts applied to optimizing our engine. Plato is our commercial stage platform optimized for enhanced potency, safety, and global scale. And last year, we took what was already a proven platform and we implemented 3 important upgrades: a new vector, an optimized vector, optimized approach to conditioning, and fully automated manufacturing, all of which contribute, we believe, to increased vector copy number, enzyme activity, chimerism, and ultimately durability. So I'll just quickly go through some of the metrics behind Plato. By pivoting to Plato in patient number 4, we see a 2x increase in multiple potency measures, such as enzyme activity, VCN, and transduction efficiency. We attribute this to the improved vector. And then the second upgrade related to the conditioning approach. And busulfan historically has been used as part of a chemotherapy regimen combined with agents like cyclophosphamide with high doses and targeting oncology patients with impaired marrow. This result is a seminal paper that's shown that using busulfan as a sole agent with tightly monitored dosing to avoid out-of-range toxicity and applying it to patients with healthy marrow, healthy livers, and healthy immune systems leads to a very different tolerability profile. So our approach to conditioning, refined as part of the Plato platform, is a single agent, single cycle busulfan, tightly monitored, and we believe that this represents real progress in attempting to improve the tolerability and faster recovery of this patient population. But there's a second benefit of going to busulfan, and this is something that's been pioneered by others and now implemented by us. Companies like Bluebird have already shown incredible curative effects above the neck in the central nervous system by applying lenti gene therapy with busulfan. Why that matters to us is here we have an MRI of a Fabry patient's brain. And if you look at MRIs of Fabry patients' brains, about half of them have tremendous white matter lesions, which translates into executive function deficit, again, what the Fabry patients call the Fabry fog. Our hope is that by combining lenti with busulfan, we have the potential to address these above the neck manifestations, which of course today's standard of care doesn't cross the blood-brain barrier and cannot address. The third and final upgrade of Plato relates to manufacturability. By implementing large bioreactors, 200-liter serum-free suspension bioreactors, so that each run with a 10 to the 9th titer can produce enough vector for 40 to 50 patients, takes the vector off the critical path and really makes it a long lead time critical raw material. Drug product manufacturing in a fully automated system where if you hit the go button of these automated pods, whether you're in Texas, California, Melbourne, or Europe, you get the same outcome, eliminating interoperable variability and allowing you to scale tenfold by having multiple pods in one clean room and potentially a low-grade clean room, really addresses scale and the ability to quickly and efficiently globalize. So a critical component, and today we are operating in this automated system in multiple continents. So the combined effect of all of these upgrades at 6 months in vivo in our first Fabry patient to receive the Plato upgrade is a 4 to 5x improvement in enzyme activity on a therapy that was already working, that was already controlling the disease. So Plato, we just think, gives us even more confidence that we are on the right track. So as of now, all Fabry patients, all Gaucher patients receive our Plato upgrades, and we can say that our engine is revved and AVRO is powered by Plato. So to conclude, we're running 4 clinical trials, 2 in Fabry, 1 in Gaucher, 1 in cystinosis. We'll give our next update at R&D Day in Q4 of this year. We've dosed 12 patients, and we can say that we're excited by the early efficacy and safety results across the platform, and we look forward to continuing to execute on these clinical programs. Thank you very much.